So we will now move over to Roy Ehling. He's a he works at Engimmune, and we're in Switzerland, and he's going to talk about synthetic coevolution of neutralizing antibodies with SARS-CoV-2. So Engimmune is a preclinical biotech company specializing in the development of safe and efficacious soluble TCR therapeutics for oncology indications. Roy has received his PhD at ETH in Science Lab, where he has worked on antibody engineering. And I think most of the work that you're going to show today is from that period, I think. Yes.
Exactly. Thank you, Victor, for the introduction. Indeed, I'm going to talk mostly about the stuff that I did in my PhD. As the title suggests, we were trying to develop more methodology regarding developing a coevolution system of neutralizing antibodies with SARS-CoV-2. Can you see my slides?
Yes, perfect.
Okay,
You can
perfect.
hear and see.
Yeah, so first, I mean, the usual background slide on SARS-CoV-2, most of you are familiar with this. COVID infections have really occurred in waves with particular variants that have some sort of novel phenotype in terms of infectivity or antibody immune evasion dominate each wave. And there is also evidence that suggests that there is persistent infections that then accelerate SARS-CoV-2 evolution, typically in immunocompromised host. And these typically herald these new waves of infections. Now, with this audience, this slide is kind of obsolete, but what I really want to highlight here is important for the later stages of the talk, and that is somatic hypermutation. We heard a little bit about it already in the previous talk, but basically B cells are in germinal synthese where they are exposed to antigen, and a process called somatic hypermutation introduces mutations in predominantly the CDRs, but also elsewhere, that then leads to an affinity maturation and clonal expansion. And I just wanted to drop some interesting papers that studied SHM in the context of SARS-CoV-2 that I found really interesting. And those have shown that over the course of the infection, first germline-like antibodies and then somatically hypermutated variants of those show increased neutralization breadth and affinity. Now, work out of the University of Texas from George Georgiou's group and others have demonstrated that you can take the serum that is predominantly a plasma cell product, as we know, contains mostly oligoclonal responses. So there's a handful of lineages that really drive that SARS-CoV-2 reactive serum titer. And only very, very few of those are actually binding the RBD, which has been found to be the primary neutralizing compartment within the serum. But what I want to show with this slide is mostly that the serum response is governed by lineages. As we just heard in the previous talk, it's not just one single antibody, but actually every time clonal lineages that contain many different variants of one antibody. Now, we were also interested in plasma cells and plasmablasts early in the pandemic, but without the know-how or machinery to perform proteomic analysis or mass spec stuff, we really resorted to single-cell sequencing of plasma cells and plasmablasts from COVID-19 patients. And because we were familiar with this, we wanted to validate those using mammalian display, sort of like combine the features of a mammalian, of a memory B cell with plasma cells. So we published this late in 2021, early 22, where we took expanded clonal lineages from plasma cells, grafted these into hybridoma using CRISPR, and then screened those en masse by fax. And as found already previously in the serum analysis study from the University of Texas, we only found very, very few RBD binding candidates. But what really is the take-home message here is that even early in the pandemic, pre-alpha variant, we found that most of these are already being escaped by alpha, beta, delta, gamma variants, whatever you name it. Like towards the end of the characterization phase, so many variants had appeared that it was clear that these variants would quickly outmaneuver any given antibody. Now, one really cool thing about hybridoma, and this has been mentioned in the prior Q&A session, is hybridoma not only display antibodies, but also secrete them into the supernatant at significant levels. So one cool thing about our platform was that we could use the mammalian display to do the facts, but then also just sort individual clones, or say just have that stable cell line already to make antibody for us for downstream assays such as BLI and ELISAs. Now, serum has been looked at by many groups. So there's a really cool paper from Alison Greeny from Jesse Bloom's lab. And what they did is they took out the RBD binding antibodies from serum and screened them on a yeast library that has a deep mutational scanning library across the entire receptor binding domain. And they found that these RBD antibodies are quickly escaped by specific positions within these RBDs. And but one drawback of this approach is that it's really tough to deconvolute which RBD binding antibodies are escaped by which mutation. So there's not really any matchup. You can't deconvolute this unless you go back to doing proteomics and mass spec. An alternative approach from Rockefeller was that they took RBDs and made a chimeric virus that then is able to mutate away while being challenged with a very defined mixture of potent neutralizing antibodies, and then found very interesting epistatic mutations that occur depending on whether it's grafted onto a wild type RBD background or a BA1 background or BA2 background over time. And our lab essentially did something similar, or a hybrid approach between these two, where we also wanted to go in with a defined antibody, with defined antibodies, but instead of going with a deep mutational scanning library, so across one NNK across the entire RBD, we went with combinatorial sequence spaces to really try and find and capture epistatic mutations and really probe a much larger sequence space of the RBD, especially in the context of Omicron, where there are like 15 or more mutants popping up at a time. Now, in this parental study, we developed a tool called deep mutational learning, and we show that we can effectively apply machine learning tools to predict ACE2 and antibody escape in RBD variants. Now, to circle back to our initial hybridoma platform, we wondered what would actually happen if we, instead of just taking a single antibody and characterizing this, what if we first do something like somatic hypermutation on it and see, because that is more reflective of how the immune response would typically function, is you have clonotypes and not just one neutralizing antibody. And what would happen in these cycles of evolution? So say you have an antibody, it encounters a variant of SARS-CoV-2, it is reshaped, and then how would it deal then with the next iteration? So we kind of thought a little bit about how could we create a system like this, sort of in vitro, and really learn how clonotypes would behave and how RBD breadth would be influenced. Now, back to the somatic hypermutation slide, we first started out by selecting a number of antibodies, and 4 of which were at the time approved antibodies, but now have been discontinued because they're being escaped by virtually any variant. So these are pre-alpha antibodies, as well as some of our own. So these MAP50, 64, 82, these are from the initial plasma cell study, some of which have really, really low rates of somatic hypermutation. Actually, this is supposed to be VL. This is a typo. Anyway, but then design consideration, how do we actually recapture somatic hypermutation? It's a process that creates so much diversity that we really have no chance of capturing that in a mammalian display platform. So we sort of decided to meet somewhere in the middle and just focus or use the unique feature that most of somatic hypermutation occurs in the CDRs. In this case, we decided to go for CDR3 and CDR2 across all 8 parental antibodies and just create libraries that were on average between 20 and 50,000 library members by creating a mutation in the CDR2 and 1 mutation in the CDR3. So really 2 edit distance, soft mutagenesis, and we call this synthetic hypermutation. So the workflow was as follows. As I just mentioned, we first generated the antibody libraries, then sorted these into distinct pools. So imagine you have a library that displays an antibody. You sort these first into alpha, beta, and delta. These would form then distinct neopools. And because of our system, we could then purify out this defined mixture of clonotype variants, have that as a protein pool, and use that to stain, again, a yeast RBD library and detect which RBD mutants would then either bind that still or escape it. Apply deep sequencing across every single step of the cycle, and then feed that all into machine learning methods to really probe the entire sequence space that we're missing just by sampling. On the left, you see exemplary crystal structures for these antibodies that are well defined. And at the time, these were the three predominant epitope groups. So they're called RBM, or just class 1, 2, and 3. And they mostly overlap with the ACE2 binding site on the receptor binding domain, also called the receptor binding motif. And in the middle, there's a sample selection of how this would look like. So this is the antibody Regeneron 33. We first sorted this antibody library for stable display, because obviously there are many variants that flat out don't fold correctly anymore or have stop codons, so those would fall out. And then basically stain this with either the RBD alpha variant or the beta variant or the delta variant, and so on and so forth, and then apply deep sequencing once this pool is sufficiently enriched. And you can appreciate, so this is just a sample, but you can appreciate how there's diversity created, where the diversity is created. Obviously, it's dominated by the wild type, because this, again, is an NNK library, so I just chose to not disable showing the wild type. But you can clearly appreciate how there are some positions that become more fixed than others. Yeah. So what I just mentioned to you is essentially the first round of evolution, so to speak. It's kind of a cheap way of looking at it, but essentially we exposed that specific clonotype, were it to exist in a germinal center, to an antigen, a repeat exposure, and it would now start to form affinity matured variants towards it. So this is the first synthetically SHM clonotype library. Now, in the next round of this evolution, we took, because by the time we reached that stage, BA1 was, or Omicron started to form. We wanted to see if any of our libraries actually bound BA1 after being exposed to just affinity selection on alpha, beta, or delta. And interestingly, some of them did. Of course, the majority didn't, but it was very cool to see this down selection from a larger mutational space to a much more focused space, as you can see here from the sequence logos. And as we expect, there should be only 2 mutations in CDR2 and CDR3. And not only were we able to show that the FACS shows binding, but also in some cherry-picked examples that we ended up producing again, we managed to recover also neutralization. So where the wild type was previously flat, you could see here in the K417N case, which is a mutation that drives beta escape, as well as in BA1, the wild types are basically flat, and then the mutants nicely recover a sigmoided shape. This in itself sort of validates as well observational studies that I mentioned earlier, with SHM being able to recover some binding and breadth, but in a way, this was a nice validation that this synthetic somatic hypermutation kind of has similar outcomes. Of course, it's a much more simplistic version than that. But the fact that simple selection for alpha, beta, and delta can create BA1 binding clonotype variants was pretty cool. Now, because we have structures available for Regeneron 33, we could have a deeper look into where these adaptations would occur. And next, of course, we wondered, like, what do these adaptations have in terms of cost? So if we look at the position, so the black bars are basically the areas within the CDR. So that's on the left axis, on the y-axis, where they would contact the RBD, which is the positions are on the x-axis. And positions marked in red are the ones that are mutated in BA1. So while an adaptation in the position 102, which nicely recovers, and I've shown the neutralization plots, nicely recovers neutralization, it only is in close proximity to other positions that are unrelated. So clearly there's a chance that these have a cost. In order to prove or probe that cost, we sort of went back to the parental study, or just thinking back to the parental study. We have all these yeast RBD libraries that are either tiling libraries, 3NK, or combinatorial libraries with a large sequence space. And in addition, in our manuscript, we generated 2 additional libraries that we call the delta and the beta gamma libraries. So these have fixed positions in addition to supplement to really make sure that delta and beta gamma lineage derived RBD variants are actually represented sufficiently in the library, because as I mentioned, with this high dimensionality or the high variability of these libraries, it might be that we end up undersampling these lineage members, and these were the ones that we were particularly interested in. Now, combinatorial mutations are really interesting for epistasis, but one drawback of making a library design like this is that it's really limited to one sort of class, and they're not cross-class. So we were hoping that we can ameliorate some of these issues using machine learning and training models. But before we reach that point, I wanted to show you some of the sample selections. So again, going back to sort of the workflow, we have these purified, very well-defined antibody libraries that were pre-sorted for a given antigen, in this case, alpha, beta, delta, or Omicron, and then stain RBD mutants that are a pool of these tiling and combinatorial mutants, and then sort and apply deep sequencing to the ones that maintain binding or are escaped by the yeast or the RBD. And of course, there's always a lot of noise and bystander mutations in combinatorial mutagenesis, but if you plot the relative frequency of the amino acids in each position of the RBD present either in the binding or in the escape fraction, you can hope to find positions that might be key to the escape in this newly affinity matured or evolved clonotype pool. And so here on the left, I'm showing class 2, which is mostly like the beta, gamma, and BA1 lineage that we're interested in. And you can see how there's dramatic shifts in neighboring positions that were not observed, say, when you go from wild type to beta. But then also completely sort of reduced sensitivity to specific positions that were actively selected for. So MAB82 previously was not binding the beta variant. So if we select for beta, you can see how the K417N mutation sort of is no longer relevant. It's not that if you were to expect that it binds it now, then they would have to appear here as letters K and N, but it just is shifted in terms of its importance. And that was interesting to observe. Clearly, there are other positions that now become more important, and that indicates some epistatic shifts that are caused by the change in the paratope. And similarly for class 1, we can also see similarly big changes, but in the opposite direction. So once we selected MAB82, for example, for alpha, beta, or the Omicron library, it suddenly starts to be escaped by the delta lineage, which denotes here the R and the K quite significantly, while it previously had no issue binding it. Now, a lot of these trends are really tough to interpret. We generate like one of these plots for every single clonotype library, and there are over 30. So it's really tough just visually to inspect this. So we thought we need to apply machine learning to really be able to not only make the analysis easier, but also to be able to go beyond things that we can find in the data and really have a more like thoroughly probed sequence space. So we applied a similar ML strategy to the parent study. So having ACE2 positively selected RBD variants, and then that using that as a mask, and then only obviously selecting RBDs that bind ACE2 and predict the binding or escape of antibodies for given RBD mutants. However, we ran into some issues when looking at MCC and F1 scores. We found that they're naturally, due to some of the data scarcity that just happens because you have a library that does not bind a certain lineage anymore, we ended up having poor performance for a number of these data sets. So Mason set himself as a goal to improve the overall architecture of this whole thing and combine some novel elements. So he was combining a protein language model with masked label modeling, as well as inter-attention transformers, which in their combination is somewhat novel, to not only help us with not generating N models, because each clonotype library would be one, but sort of make a unified model, and especially help with this data scarcity and data imbalance. So we formulated 3 different learning tasks. I mean, this with the desirables or deliverables for this is to be able to show good predictions when trained on low edit distance, be able to show good predictions when trained on high edit distance, and also to perform well when holding out specific positions during training. And neuromutants such as the JN1 and BA286 variant, they have really high mutational load. And we really wanted to hone in on this and show that the data that we have generated and the model that was trained is able to perform well on tasks that were tough, given the data, given the input. So the bright green bars are our PLM. It performs basically across the board a lot better than any of the individual components, as well as the CNN that was used before. So most of the values here are MCC above 0.7, which in the field I think is considered pretty respectable. But also on the much more difficult learning task, which is the hold out positions, where you basically just remove one position and then try to predict on it. It's also performing moderately well, but a lot better than any of the other components or other models. And that is across when you fraction or stratify the data into the full data set, the top labels or the more scarce labels, it still performs reasonably well, almost scratching the 0.7 mark on the most noisy and scarce data. Attention is all you need. We wanted to understand a little bit more what the transformer was actually looking for to make these classification decisions. So in the context of BA1, you can plot essentially the relevance of each position for each data set, and by far and large, positions 499 and 93 seem to be the most salient, but other positions as well. Yeah, indeed, many of these indicated positions have been found to or identified in convergent evolution of SARS-CoV-2 that confer like improved ACE2 binding, or also contribute towards immune escape. But yeah, particularly the ACE2 affinity enhancements are interesting always, because there have been papers that showed that ACE2 affinity enhancement is crucial to enable other positions that usually would be deleterious in an epistatic fashion to then lead to antibody escape. When looking at the embeddings of these antibodies, we were actually pretty surprised. Some of the embeddings seem really unexpected. So where the sort of clonotype libraries shift away dramatically from their wild type, like here, but then of course, you can always cherry pick with these dimensionality reduction techniques and say, oh, here I see something that makes sense. Yeah, when we want to look at how much of our clonotype library is binding throughout the entire RBD sequence space, we actually see, or the model performs the same or recapitulates trends that we observe biologically as well in terms of library breadth. So when we do a FACS experiment, we can see whether more or less is ending up in the binding or escape fraction, but not like what. And using our model, we were able to really comprehensively map which mutations are escaped and bound by the clonotype libraries. And we also observe the trends that we see by FACS is that, for example, when you select our Regeneron 33 antibody for beta or Omicron binding, then its breadth is improved, while conversely, when, and I touched on this in the neutralization, when you sort or mutagenize MAB82 and select it for beta and Omicron, it becomes a much more nuanced or limited breadth. And we were sort of glad to see that the model produces similar results as observed by FACS, which sort of validated that the model is not learning. With ML, you always have to be careful that you're not training on BS and then something weird comes out. To summarize, we took 8 antibodies, partially from the clinic, partially from our own discovery efforts during the pandemic, and generated clone-type libraries, developed a somatic hypermutation clone, and stratified that into different sublibraries, depending on whether they were affinity matured for alpha, beta, or delta variants, as well as Omicron, to produce a large number of data sets that contain single mutants, including within germline-like antibodies that were able to recover neutralization against BA1. And again, these were all like pre-alpha discovered antibodies. Then, together with our parent study, we developed 3 combinatorial and 3 NNK tiling libraries that also represented delta and beta gamma lineages, and we sorted all of this for ACE2 to make a functional library, and then determined these binding and escape patterns from those clonotypes to really do the breadth analysis, and this breadth analysis really shows how escape patterns can be shifted dramatically from their unmutated monoclonals. And lastly, Mason created a unified model that we term RBD-PLM that is able to accurately predict on unseen RBD mutants, as well as probe a much greater RBD sequence space. And this model was validated with a whole plethora of performance metrics that you could see in the paper if you're interested, that can generalize to a high mutational load and recaptures biological trends that we observe. And with that, I'm a little over time, but I want to thank particularly Mason and Max, who was working with me on this project, and also our PI, Professor Reddy, and our collaborators who did a lot of the neutralization studies at Karolinska with Ben Murrell and Dan, and my funding sources. And I'm happy to take any questions.
Thanks, Roy. Really, really nice talk. Are there any questions? If not, I have some questions. And I will start asking them. If on the one accuracy slide where you had the bar chart with all the PLM and CNN and so on, what is the accuracy? Is it for, okay. Now I see on the left is the edit distance 3, and then you predict 1 and 2, and then you have the
Exactly. Yeah, so the
and
task,
make
it's not edit distance 3 necessarily, so it was trained on edit distance 3 RBDs.
Okay, and then you predict the lower ones.
And then you predict in the lower range of mutations, so like 3 or less. And for edit distance 10, sorry, I should have said that. So with edit distance 10, you train on this edit distance, and then you predict on edit distance 11 through, I think the threshold was 23. And it still does that pretty accurately. And this is for a subset of antibodies.
That's interesting. And did you
Based
try,
on-
so how did you choose the 3 and the 10, just arbitrarily, or did you
Just
try
arbitrarily,
different?
because the average of the mutations in the 3 and NK is something like 2 point something. And the edit distance for the combinatorial is shifted to more like 8 or so. So it was arbitrary mostly.
That's really interesting. And did you also try to kind of you know kind of cluster the data on the training a bit differently, like using only very sequence similar or very sequences similar based on not only the edit distance but also other things, or it's or mostly
Not that I'm
only
aware of. I would have to
yeah.
check with Mason what else he tried. I think he used different. So what he did was he used for sure many, many different seeds. So
Mm,
trying to check different subsets and see
yeah,
if the performance to hold. So I think the data represented here are at least 5 seeds.
yeah.
Um, Whether he tried it on a different set of antibodies, I would have to check. I don't know off the top
Yeah,
of my head,
it's
but
not
I think
problem. I
Nathan's
think
in also in the
yeah,
talk. So if you're really curious, you can have a
yeah.
chat with
No,
him.
I think it's not. Yeah, yeah. No, I have another. So you showed some structural kind of where you translate what you find in the sequence level to the structure level.
Mhm.
How extensively did you do that with your findings? And is that structure experimentally or is that modeled then? Or yeah,
So
so yeah,
yeah. This is one thing I'm a little bit sad about, or is still like in the works. I mean, this is still in revision.
yeah,
I'm trying to generate. The problem is that for recovering Omicron binding, we only have the 2CO8 antibody that
yeah.
was already known to bind it a little bit, and out of the antibody libraries, only Regeneron 33 and this MAB 82 was able to really show good binding. We also see it a little bit in MAB 64, but that wouldn't have helped us because neither MAP 64 nor MAP 82 are, we don't have a structure for it. I
Mm,
did attempt to use some Rosetta, for like Rosetta app and docking to figure
mm,
out the position, but for me as a layperson, when it comes to Rosetta, it's really tough to evaluate which one's a good model. I am still
mm.
planning to try it for a couple to do it as extensive as I did for Regeneron 33, but this is all based on things that were published.
Yeah, got it, got it. Jamie or Felix? We don't know who's
Oh
there.
yeah, it's Jamie.
Yeah,
Really nice talk, Roy. Thank you so much. Yeah, I wanted to ask you about the affinity ranges when you're for your uh, uh, synthetic RBDs that have never been seen before. What kind of affinity ranges are you pulling out of your optimized libraries? And also, you
So,
probably mentioned it and it just I missed it. What kind of neutralization numbers are you getting? Um, do you
yeah.
go back and do for the a particular RBD, are you doing neutralization assays and that kind of thing?
So if I understand, so let's start with the second question. The neutralization for the recovered variants is of course nowhere near the wild type. I'll just go out, go out and say that, but it is still, we get sigmoid curves, but of course they're still like 10 to 100 fold lower than what the wild type used to be. Um, and that is a result. But we have to keep in mind that these are just single mutations, and it's also not exactly clear how what neutralization capability will be necessary, like in vivo, to actually confer some sort of um, selection to then be
Right.
recruited more into germinal centers and continue on the evolution. I mean, in vitro, you can really essentially, it's called a round, but what we do is just a very small snapshot in time within that germinal center of like one extra mutation that confers a little bit better binding, and then immediately snagging it out and testing it. So it does not really recover, of course, as it would in vivo. And then your first question of the affinity, was it more related to the antibody or the RBDs?
What I was wondering is when you screen your optimized library, I can't remember the name of it, uh, then with a
You mean
given
the in silico library?
RBD, and you get out binders from your sorting. I was just wondering what the you make the FAB and you do affinity um,
Okay, so the antibodies themselves.
analyses. I don't know with
So
SPR or something like that? What kind of numbers are you getting for, affinities?
yeah, so we haven't determined the affinities of a clonotype library. I believe they would be really messy if one tried. Um, but yeah, so what we
Yeah,
typically do is we just use the wild type as the benchmark when we stain. So, okay, let me just pull up the slide. In this case, for example, right? So you would stain first with a wild type antibody, um, and just see where that wild type would sit in terms of binding and escape. And then you would draw gates around that, and you stain the wild type and your and your antibody diversified clonotypes at the same concentration. So things that bind lower than the wild type are effectively escaped, and things that bind the same are considered to be maintained. It could be, of course, that you stain within such an access that the nuance is a bit less defined, or that you might effectively have something that would maybe be slightly escaped, but you still consider it binding. But yeah, that's the only thing you can do if you didn't mean to produce hundreds of clones and check their individual affinity. I don't think we've tried, but I can imagine that trying to determine the affinity from a polyclonal mixture might be tough by
yeah,
BLI.
right, yeah, but I guess you can't get clones out of there, sure. Okay, thank
I
you.
mean, we could by single cell sorting our hybridoma, which we then did, and produce individual variants. If I go back to this slide, so these were individual variants that we single cell sorted, and then used the purified product to do neutralization plots. But we just went straight for neutralization. We didn't want to. I mean, affinity is nice, but neutralization is really more important. In that context, I believe.
And maybe one last question before we have to close because we are running over time. But so all the things that you're showing is synthetic, which is great, of course. But then because you're calling it co-evolution, right? So I'm thinking about if all of this had happened in a mouse or in a human, and you were referring a bit to known antibodies before and after, and so that were from public sources and that were maybe also found in humans. Um, the antibody sequences that you are finding, are they if you were testing humanness or something like how natural are they in terms of would they be immunogenic, things like that? Yeah, from an antibody engineering therapeutic point of view. Yeah.
I mean, I haven't evaluated this. I don't think there's a good way to evaluate immunogenicity that has really been proven or tried and tested.
But at least humanness can be done in terms of how close, so how likely they would be to be BDJ recombined and then also having certain somatic hypermutation or something. I think that could be done if one wanted to. But you
I
guys
mean,
didn't do it.
I,
Yeah.
I could
Yeah.
look into it. I haven't done that analysis.
Mm.
Presumably, at least when it comes to somatic hypermutation, given that we really focus like right in the middle of the CDRs, at
Should be
least with
fine.
CDR3, I'm not too worried.
Yeah.
CDR2, given that it's more like framework,
Mhm.
and defined by the V gene, you have a point. I think it might be valuable to check if similar CDR2s have been
Mm.
observed,
Mm.
particularly by the same germline. So I mean, if we look at this guy in particular,
Yeah.
MAP82 is a germline like antibody. It has 0% SHM. The question would be if that's a good point. I should look into whether mutations in positions 53, 54, for example, have been
Mm.
described in the past.
Mm. Mm.
Um, yeah,
Yeah.
so it's a good point.
Great. Um, Unless there are further questions, I think we have to close now. Thanks again, Roy, for the beautiful talk.
Thank you
And
for the invite.
we will be back with our seminar in January 30th, I think, 2025, new year. Yes. Thanks, everyone. Bye-bye.